honored to be able to be with you all today. And I'm especially thankful for the organizers who have done a wonderful job in keeping the intermediate filament community together. And those are Rebecca in Zurich and Emma in Paris and Oscar, who's in Turku in Finland. So today I'd like to tell you a few things about our work and what we're in the middle of doing right now, and also a little bit of history will be mixed in to give you a longer term perspective. But the topic of this meeting, connecting intermediate filaments, is just perfect because actually on the first slide you can see I said intermediate filaments networks do connect the nucleus to the cell surface. So let's get on with the slide. So let me just review for you. Let me see if I can. I'm not very good at zooming, but I'd like to start towards the cell center with the nuclear lamins and just tell you a little bit about the background that led us to do the work that I'm going to tell you about. And that it really started in terms of understanding how the lamin meshworks inside the nucleus interact with each other with our work, early studies on progeria. And it really was the misshapen and, as Francis Collins says, the lumpy, bumpy nucleus of the progeria patient cells that provided us with some important insights. So as you may or may not know, I'll just quickly review it for you. Progeria is caused, it's a premature aging disease of children. It's caused by a single nucleotide substitution, 1824 C to T, which is a silent mutation, G608G. And this is it's a heterozygous situation. And in fact, what we were able to show years ago as the truncated protein known as progerin, which is the nuclear lamin A that's mutated in progeria. As this progerin is expressed over a period of time in cell culture, we found that it accumulates going from an early passage culture to a late passage culture. And as the accumulation of progerin, which you can see here in this middle band, this is lamin A, this is lamin C. Progerin is towards the middle. This is a permanently farnesylated form of lamin A that causes the nucleus really to alter its shape, and it also causes a loss of peripheral heterochromatin, which I'm showing you on the right as a loss of the inactive X chromosome in a female control cell and a female patient cell. We showed that the chromatin actually was released from the nuclear surface. And here in the electron microscope, you can see that peripheral heterochromatin on the left in progeria is missing. But one of the most impressive things for me, being interested in lamins for many years, was the blebs that form, these large protrusions, have lamin A and lamin C, but not the B-type lamins. And as you know, there are 4 lamin isoforms. Up until that time, we always thought they were, more or less, we thought they were always together and they always formed sort of a heteropolymeric polymeric network, but they never really separated from each other. So this A and B separation was the first clue that something was going on with the formation of lamin networks inside the nucleus. So, and remember, the lamina itself is only a relatively very thin layer, about 14, 15 nanometers thin at the nuclear envelope surface, and packing 4 different lamins into this structure is really quite amazing. And we know that the lamins represent the major protein of the nuclear envelope surface. So we started asking questions about how these networks actually form in wild type cells. And I should tell you, we've taken advantage of cells that were provided to us by Yixian Zhang. Just to let you know that she has provided us with knockout mouse embryo fibroblasts for each of the Lamin isoforms. And we have also mapped the Lamin isoforms using very specific antibodies using now structured illumination microscopy. So when SIM came along and we could do 3D super resolution, we started asking questions about lamins A, C, B1, B2, and how they assemble with each other. And in this way, we were able to, using computer vision techniques, which were developed in our lab by Mark Kittisbokel, but actually started this work when he was in Kulu Jacquemin's lab at the University of Texas in Dallas at UT Southwestern. And using a steerable line filter and applying threshold and connectivity analyses, we were able to identify each of the lamin fibers. And we could even identify, let me see if I can minimize this. We could even identify structures that comprise the meshwork, so that the fibers which surround each of the spaces, as you see, where we call it, we say that's a face, and the fibers themselves are the lamins, and then they form junctions. So what they form is irregular meshworks of polymer at the just below the nuclear surface. And each lamin we were able to show forms a separate yet overlapping structure with the other lamins. So, and just by definition, I just want to mention to you that we call fibers the structures that we see in the light microscope using super resolution microscopy. And then a few years ago in Ohad Medalia's lab, Turgay in a paper that we worked on them with them on that was published in Nature, well, we discovered that in fact the lamins, rather than being bona fide 10 nanometer intermediate filaments, were in fact only 3.5 nanometers in diameter. Their length was about 400 nanometers, and their persistence length, which was very short, 50 to 2,700 nanometers, relative to actin, microtubules, and even Vimentin in the cytoplasm. This is a very short persistence length, which says that it's probably a very flexible polymer, and we know that this flexibility is important in understanding the mechanical properties of the nucleus, which has become a central topic in cell biology. Just at the bottom here, you can see some individual lamin filaments interacting with the nuclear pore complex in a cryo-EM tomogram, cryo-EM section prepared by in the Medallia lab. So we've started, and this is going on now, to map the interactions between lamins and other major components of the lamina. We've started both with nuclear pore complexes and peripheral heterochromatin, but today we've advanced much further in our understanding of nuclear pore complexes and how they interact with the lamina. And so we've chosen two major structures that interact with the lamina to begin with, and that is the nuclear pore complex and peripheral elements of heterochromatin. But today I'm going to tell you mainly about the pores. And throughout these studies, I should mention that Yixian Zhang and members of her laboratory at the Carnegie Institution have been terrific collaborators and have provided us with many cell types, including knockouts of each of the lamin isoforms for our studies. So that, what we found was, if you actually look at a wild type nucleus and you try to define a relationship between, in this case, nuclear lamin A and the nuclear pore complex, as stained in this case with the antibody against the nuclear porin it's called 414 antibody. You can see that it's very difficult. The green fibers are the lamin A fibers and the pores, but it's so crowded and so packed that we had to develop a trick. And the trick was really developed for us by one of Yixian's lines, and that is the Lamin B1 knockout line, which turns out to give an expanded mesh of Lamin A. So you can see the Lamin A fibers here, but notice that now that the pores in magenta are actually lined up. They actually move with the expanded mesh. So they line up with the expanded mesh. And since then, we've, and I should say that I'm not going to go into a lot of detail on this because there's far too much to give in this talk this morning. But there is a paper now in press, and will be out in April in the Journal of Cell Biology. And this was done, most of the work was done by Mark Kittis-Bokuku and also by Takeshi Shimi. Takeshi is now in Tokyo, but at the time he was in my laboratory. In fact, continued the work, helped us finish the work while he's in Tokyo. What Mark did was to define, using subpixel analysis and various types of computations, the center of the lamin fibers in any given section using structured illumination. And he could define this by subpixel analysis, and then also could define the center of each of the pore complexes, which were a small round dot, but he could determine the center of that spot. And what we found is that consistently the lamin fibers are approximately 30 nanometers away from the pore. So it's not, we never see them actually touching or overlapping. And the center to center spacing between the center of the fiber and the center of the pore is about 114 nanometers. And you can now, once we learned how to do this in the expanded meshworks of knockout MEFs, we actually have been able to do it in wild type MEFs. And without going into great detail, because it's far too much work, you can look. The paper is also in bioarchives, but should be online pretty soon in the Journal of Cell Biology. What we did was we analyzed many 3D sim data sets with 10 to 20 nuclei for wild type and all the lamin knockout MEFs. And basically, we could monitor the distance from lamin fibers to the center of a pore, center of fiber, center of pore, and the distance from the face. I defined the face for you as the area surrounded by the fibers, the distance from the face center to the center of the pore. And using this bivariate analysis, we're able to show and conclude that lamin A and lamin B1 fibers have a more obvious and pronounced relationship with nuclear pore complexes than either lamin C or lamin B2 fibers. So this so this told us that the lamins, which we knew already from electron microscopy, but we weren't sure of which subtype of lamins was closely approximating pores. So now we've begun to actually map each lamin relative to pores, and then we thought we would start looking at pore complex components. And here, in a further collaboration with the Medallia Lab and with Melton Tatley and Ohad, we've been able to show that the lamins, lamin fiber, lamin filaments in the electron microscope using EM, cryo-EM tomography, actually associate with the inner, quite frequently with the inner ring structure. And this is true for both lamin A and B using immunogold labeling and cryo-EM. And because the inner ring structure contains certain known components such as Ellis, NUP153, and TPR, what we did was to go in and silence each of these components. And in summary, without giving you again all the details, which are going to be in this JCB paper, we can show that the lamins or that Ellis, when we silence Ellis, the pores seem to come off and cluster in the center of the face. So they come off the fibers and cluster in the center of the face. To some extent, we also see this with NUP153, but in fact, let me see if I can see this here. In fact, most of the association appears to be with Ellis, which we know is a primary component in assembling the nuclear pore complex. So in this way, we're mapping the interacting sites, structural interacting sites between pores and nuclear lamins. And this is the cartoon we made for the Journal of Cell Biology. They wanted a cartoon at the end to summarize all the data. And basically, what it says is that lamin A or B1 filaments in the electron microscope, fibers in the light microscope, actually interact with pores, and a major linking component is Ellis. And that when we silence Ellis, we actually can displace it from the pore complex. Under normal conditions, when we look at an expanded meshwork, we find that the lamin A fibers in a lamin B1 knockout, that the pores follow the fibers, which for me at least indicates that the lamins play a major role in organizing the nuclear pore complexes. I should also mention to you, because I find this quite intriguing, let me see if I can go back one slide, that the mesh, when we silence Ellis, that the meshwork of lamins expands, so that the meshwork size increases, and which says that the interactions work in both directions. So we see both lamin pore interactions, pore lamin interactions working together, and they seem to be able to control each other's architecture. And then I just want to mention this too, but I'm not going to say much more. This is now going into a more extended study with Yixian Zhang. But what we did find, if you look at chromatin, look at DNA, in this case, lamin B2 in the upper left, and a peripheral heterochromatin mark, in this case, H3K27 trimethylated. It's really a remarkable meshwork of, because this is a SIM image. It's a relatively thin focal plane at the nuclear surface. It's just packed with DNA and lamins and of course, pore complexes and many other components. But what we've been able to show initially is that when we silence LB1 and expand the mesh, we use this trick again, we find that the chromatin fibers at the edge seem to follow the fibers of the lamins, indicating, again, a close relationship and the possibility, of course, that lamins might be regulating the organization of chromatin at the peripheral region of the nucleus. And I should also mention that in these, again, in the expanded mesh of an LB1 knockout cell, MEF, that we also could see acetylated histone inside the face areas. So whereas the heterochromatin lines up, the euchromatin tends to be towards the more towards the center of the face. So we're hoping in the future, in the not too distant future, to begin mapping in detail which lamin-associated domains of chromatin interact with specific lamin isoforms. And that we're doing with Ishii and Chang. We've also started to look at whether lamin, because there's a lot of interest in the lamina and nuclear mechanics, so we've started to look at lamin isoform, specific isoforms and nuclear mechanics. Most of the work that's published in the literature has been done with nuclear lamin A. We've extended this to look at each lamin isoform, again, taking advantage of the knockout cells that Ishii has provided. And Amir Vahabakashi, who's in my lab, who's a biomedical engineer, an expert in atomic force microscopy, has developed a technique using AFM round tip and AFM sharp tip probes. And when you take the AFM round tip and do nano indentation above the nucleus of a cell, you can get, we now know we can get nuclear mechanical data. And if you do it on the cytoplasm between the nucleus and the cell surface, we get cytoplasmic stiffness data. And you can see for each lamin isoform that's knocked out, we get a different different stiffness of the nuclear surface, and we also see differences in cytoplasmic stiffness. And I can tell you over here on the right, we're using Amir is using a sharp tip AFM probe. And when you use a sharp tip, he's been able to demonstrate in a paper that appeared in the Biophysical Journal in 2019 that the sharp tip probe actually measures the cortical stiffness of the cell. And again, we can see correlations between the specific lamin isoform and differences in cortical stiffness. So this says that lam is also influenced in some way the cortex of the cell. And we also showed by transwell migration, we're showing this now that in fact, different lamin isoforms give us different rates of movement. So when we knock out lamin B1 or lamin B2 or lamin AC, we get differences in terms of the number of cells that move over a 12-hour period from the top of a well to the bottom of a well. And we can also look at, and I can tell you, I'm not going to show you the data, but it fits beautifully with all the other data that we have. We're working with Ming Gao, who's at MIT, who's an expert on laser tweezers, laser trapping, and we're using laser trapping, beam microreology to begin to push on the nucleus and understand its mechanical properties in live cells. And I can say that the data so far that we get for all the lamin isoforms fits beautifully. And finally, we've been looking at traction force microscopy in collaboration with Chan Park and Jeff Fredberg, who are at Harvard, and also Dave Waits. And we can show that the contractility of cells is also somewhat lamin dependent. So even the cell surface, the way it interacts with the substrate, is influenced by the nuclear lamins. Just to tell you that we're now looking at the possibility that the linkage between the lamins and the cytoskeleton and the cell surface through the intermediate filament network, we think, especially Vimentin, looking at elements of the link complex. And this we're doing with in collaboration with Gant Luxton and Greg Gunderson, who are at Columbia University. And this is being done by Amir right now, and the preliminary results are quite intriguing. We're looking at nesprins and also the sun domain proteins, which of course link between pores and between the nuclear surface and chromatin. let me just switch now, because I've told you a lot about this meshwork of lamins, which forms a type of cage around the outside of the nucleus, but inside the inner membrane of the nucleus. Here, I just want to remind you, or if you didn't know it, tell you that typically in many cell types, and for many different kinds of intermediate cytoskeletal intermediate filaments, here we see Vimentin. There is a cage that forms around the nuclear envelope, around the outside of the nucleus. And sometimes it's very elaborate. Here, upper left, we see Vimentin and the nucleus of a BHK21 fibroblast. Here we see another elaborate cage in a different cell type, but always it's a three-dimensional cage that goes from the bottom to the top of the nucleus. So in collaboration with Paul Janmay and Alison Patterson, who's now a professor in physics at the University of Syracuse, and Paul, of course, is at Penn, and Amir Bahabakashi in our lab, we've been able to demonstrate that the cage of Vimentin is critical in many aspects of nuclear morphology. For instance, the shape of the nucleus is influenced by the Vimentin cage on the outside. And we do this by using Vimentin wild type MEFs and also Vimentin knockout MEFs. And we could show that the shape of the nucleus is significantly altered when we remove the cage of Vimentin, and when we put it back by rescuing, it goes back to the normal flattened, elongated shape. And then again, doing nanoindentation above the nucleus with a round tip AFM probe, we can show, Amir was able to show that the Vimentin null cells are much less, the nuclei of the Vimentin null cells are much less stiffer. And the rescue also worked. This was actually published last year, well, 2019, in the Journal of Cell Biology. So I'll just review it quickly. We also show that the loss of Vimentin increases the rupturing of the nuclear envelope, both in 2 dimensions. When cells are just sitting on a glass cover slip or a plastic petri dish, they tend to form these blebs and they rupture. And we can show that the percentage of cells, the nuclei that rupture in the Vimentin nulls is much greater than wild type. And we can also show that in 3-dimensional cultures, the situation is the same. And rupturing is determined by the loss of GFP NLS tag, which accumulates in the nucleus. When the nucleus breaks or ruptures, this spills or leaks into the cytoplasm. So our conclusions from this are that the Vimentin intermediate filament cage regulates nuclear shape and volume. The loss of Vimentin leads to increased nuclear envelope rupture when migrating through constricted spaces, which I didn't mention, but we also did transwell migration assays. And the loss of Vimentin increases nuclear blebbing and DNA damage through DNA damage repair after migrating through confined environments. And also we know the compressibility of cells and their nuclei is significantly altered when Vimentin is not there. And we see under 80% strain, as you can see here, that many more cells die of necrosis, as indicated by propydium iodide staining, than wild type. So Vimentin seems to play a major role in regulating nuclear mechanics in addition to the role of the lamins. There we go. We also noticed that Vimentin contributes to the mechanical properties of the cell cortex. And this was originally published a few years ago using sharp tip probes, which I mentioned earlier, actually measure the stiffness of the cell cortex. And on the left here, we see the results with a sharp tip AFM probe with Vimentin wild type fibroblasts, and then null fibroblasts obtained from the Vimentin null mouse. And you can see a great decrease in the stiffness of the cells at the cortical region. And we also showed using traction force microscopy that there was also a significant reduction in force exerted by the Vimentin nulls on the substrate. Okay. So this led us into looking at the potential interactions between Vimentin and actin at the cell cortex. It's been thought for many years that actin is the major component of the cell cortex, and that the cortex is regulated primarily and almost exclusively by actin, but I'd like to convince you that that's likely not to be the case. And this is an electron micrograph that was taken by Kathy Green, which she was a member of my laboratory many years ago, and well, 1986. And what Kathy saw and what we noticed actually on and off for years is that intermediate filaments really are very closely related to stress fibers at the cell surface. This is a thin section at the very bottom where the fibroblast actually contacts the glass cover slip. Now with SIM imaging, using super resolution microscopy, we can actually show that actin stress fibers, which are in red, and Vimentin interact in many different ways. And this is being done extensively now by Suganya, who's in my laboratory, and was originally started by Anne a few years ago. But we can show in green, you see Vimentin, in red, you see actin. You can show that there are many interesting kinds of interactions which suggest that Vimentin can wrap around stress fibers and maybe even enter inside stress fibers. But the only way to really show this was with Ohad Medalia. And he and myself and Dave Waits are now collaborating, finalizing a paper on Vimentin in the cell cortex. This is an example of the kind of correlated light in EM that Miriam Weber and Ohad are carrying out in Zurich. And you can see this is a cell growing on a gold grid, and then it's processed by rapid freezing for cryo-EM tomography. And the green arrows show you Vimentin at the surface, but also you can show that, in fact, the Vimentin, which here is seen in purple, and the actin is in yellow. You can see that the Vimentin interdigitates and wraps around the outside of stress fibers, frequently even lying closer to the cell surface than the actin comprising the stress fibers. Miriam also measured the distance between the actin filaments and Vimentin within stress fibers, and showed that on average, the distance was only about 11 nanometers apart, which indicates that they're actually very, very close together. It's hard to think of a known cross-linker that could fit into that small space. And there is evidence in the literature that the C-terminus of Vimentin can interact directly with actin. So maybe this is really the case. And then I should mention this, and again, we don't have time to go into all the details, but it's being worked on now, and papers are in the works on with the members of Dave Waits' lab, Helen Wu and Yinan Shen, on mixing Vimentin and actin. And Yinan actually has even managed to make mixed polymers of Vimentin, actin, and microtubules to begin to look at the properties of these mixed or composite polymers. in vitro and measure their microbiological and mechanical properties and physical properties. I think that, and I can also mention that Helen Wu has some really neat data on the role of Vimentin in looking at stress fiber dynamics. Using fluorescence recovery after photobleaching, she shows beautifully that the actin dynamics are strongly influenced by the presence or absence of Vimentin, again using Vimentin null cells, which have perfectly normal stress fibers. Okay. And I can tell you the regulation is such that with Vimentin present, fluorescence recovery is faster than it is without Vimentin. And then I could just mention that defining the high resolution structure of Vimentin now is more essential than ever for understanding their function. And I'm not going to go into details on this, but one of the nice things that's happened over the past few years in our collaborations with Ohad Medalia and now Matthias, Eibauer, who's in his lab, is that we've been able to take advantage of an old technique that we developed a long time ago on making intermediate filament enriched cytoskeletons. This is actually a cell. This is a chicken embryo fibroblast grown on an electron microscope grid. And this was our first procedure for isolating Vimentin from cells, intermediate filament proteins from cells, back in the 1970s. And what you can see, this cell was extracted with high salt detergent, DNase-1 to get rid of chromatin. It also turns out to help disassemble the actin. And all done in the cold with some calcium so you get rid of microtubules. And when you do this, you get an IF-enriched cytoskeleton. And Ohad, by rapidly freezing these, has been able to get very straight runs, Ohad and Matthias, of Vimentin filaments. Here you can see in the upper right. And the reason that it's important to have these straight runs is so that they can take short segments. And they've taken hundreds of thousands of them now and started putting them together. And just sent me some images the other day of what Vimentin is going to look like in a few months. Paper is in the works and almost done. My color blindness doesn't allow me to define all of the structures. I think Ohad did this on purpose because I can't really distinguish green and red and blue when they're all together like this. But nonetheless, one of the really exciting things is that rather than what we thought was a 4 protofilament filament wall, it's actually 5, meaning that there is likely to be 40 protein chains making up the wall of a typical Vimentin filament. And remember, this is a cell, it's not bacterially expressed intermediate filaments. It's in situ in the cell. And many of the filaments, not all, also have a central density. So I'm not sure what this means yet, and I don't think either Ohad or Matthias understand what this means yet, but it is intriguing and very, very interesting. And of course, without structure, we'll never really understand how Vimentin contributes to the mechanical properties of cells. And just in summarizing, I'd like to say that all of this that I've told you, all of the cellular data, the structural data, I think will help us to explain the Vimentin null mouse. And this is just a cartoon that was made by, prepared by John Erickson. And although the Vimentin null mouse was originally introduced as in 1994 as a phenotype less, had no phenotype, this caused a lot of grief in my laboratory. I've been studying Vimentin for so many years, and we said, oh, wow, the mouse lives, it reproduces. But in fact, over the years, it's turned out this is a very abnormal mouse. And this just indicates all of the organ and tissue systems that are affected in the mouse. And the accumulating data from cell biology will be, I think we'll be able, with this accumulating data at the cellular level, both mechanically and physically, and the fact that Vimentin can interact with, we now know, of course, extensive arrays of interactions with microtubules and with actin. I think much of this will shed light on the many, many defects that have been discovered in the Vimentin mouse. And I should mention, they're listed here. I don't have to go through them, but there are lots of them. Wound healing is one major defect. The work on that was pioneered by John Erickson, and there are many other intriguing defects. They don't kill you, but they don't kill the mouse, but they sure make the mouse subject to various illnesses and malfunctions. This is work that's being done. I should acknowledge all the collaborators here. This Karen Ridge, John Erickson, Milos Pekny, Ed Kuzmarski, and myself are putting a review together for Genes in Development. And hopefully, this has been a work in progress for some time. Hopefully, it'll be done in the next few weeks. And finally, I just take a slide from David Waits's lab, which shows you the effects of cytoskeletal. It's great. He's a physicist, and he tends to do things very in a very straightforward way. And basically, what he says is when you mess with actin, the cell usually dies. When you do something to microtubules, really without them, the cell dies. But with intermediate filaments, you can still live, but you really have aberrant mechanical behavior and a large number of diseases. So with that, I'll end. I'd like to thank everybody who's contributed to this work. I should especially acknowledge all the people in my lab presently there. Mark Kitasopokal now is at HHMI at Janillo Farms. He did a lot of the imaging work that we've been doing for the last several years. Steve Adam, I can't say enough about Steve. He's been a wonderful collaborator and colleague for many years at Northwestern. And these are all our collaborators. I'm just listing them here. Without them, and in fact, without the multidisciplinary approach we've adapted, I think we would know much less about the intermediate filament networks, both in the nucleus and in the cytoplasm. So with that, and I'd like to, of course, thank all of our supporters, and I'd like to thank all of you for attending. I think you're attending this lecture because it's a little bit less stressful than watching CNN News and the second impeachment of Donald Trump this morning. So with that, I'll stop, and thank you for the honor of giving the first lecture in this series. So.